Hi, thanks for coming. Today I'm going to be talking about some new features in QuPath and beyond QuPath, that are hopefully going to make it easier for Python programmers to interact with QuPath in hopefully exciting ways. So the material that I'm going through today is available on the GitHub link. I've just pasted in the chat. Um, There's instructions there for how to run through the notebooks that I'll be going through today yourself. There's an example project in the releases, and you'd also need a QuPath release candidate, QuPath version 0.6 release candidate 3, which has a lot of exciting new features. But probably isn't the best thing to be basing all your analysis off of, because we've still got some sort of kinks to iron out before we make the final release. So having said all that, I will share my screen. Yeah, and that's the repository that I was talking about, which includes a couple of notebooks and short scripts. And under the releases tab, there is a example project that contains a few images that we will be going through. And so I've just got a JupyterLab instance set up to go through these notebooks. So the first thing that I'm going to talk about is QuBaLab, which is a Python package that we've just released on GitHub and should be coming to PyPy fairly soon. We just have a few kind of technical things to iron out first. And this is a Python package for quantitative bioimage analysis, sort of especially in combination with QuPath, but not necessarily. And yeah, the name comes from Quantitative Bioimage Analysis Laboratory. So reminiscent of QuPath, but not necessarily related to pathology, and I guess in this case also not necessarily restricted to QuPath. So why would you want to use QuBaLab? So it's not QuPath, but hopefully they're good friends now and into the future. So QuPath is a user-friendly Java application for bioimage analysis, has some nice features for handling whole slide and highly multiplex images. But we kind of recognize that a lot of bioimage analysis research is done in Python, especially things like deep learning, and this can be hard to integrate with QuPath. And so QuBaLab is basically trying to help with this by giving us more tools to exchange data between QuPath and Python, without directly depending on QuPath and Java. So you don't need QuPath to be installed to use QuBaLab, although there are some nice features that you might want to use alongside QuPath. So it doesn't share any code with QuPath, but it uses a lot of the same conventions for accessing images and representing objects, especially using GeoJSON. And we're using the same custom fields for things like measurements and classification. So exchanging objects and data between QuPath and QuBaLab is fairly straightforward. And so the astute amongst you might recognize that there's already a Python library for interacting with QuPath, which is Paquo. And we think this is really great, and we're not at all aiming to replace it. There's kind of sort of three differences that we've noted that, if I can foreground myself, that three. Between the two, the first is sort of target audience. So Paco is mainly written for Python programmers who need to work with QuPath data, whereas QuBaLab is mostly for QuPath users who want sort of a similar API in Python and who want to be able to dip in and out of Python sort of for short stints. And the second one is sort of convenience versus efficiency. So Paco is based on JPipe, so it's kind of running in the same process and changing data that way, which is much more efficient, and it gives you much more full access to Java objects and provides some nice sort of wrapping around Java objects that we don't really provide. whereas QuBaLab is based on Py4j, which communicates data over WebSockets, which is kind of more programmatically convenient in a lot of ways, but it's much less efficient, especially when it comes to transferring large amounts of data. And the third is pixel access. So Paco is mainly for working with projects and objects. And as far as we know, accessing pixels is beyond the scope. Whereas QuBaLab allows you to request pixels as either NumPy or Dask arrays. And it also provides some functions to convert between thresholded images, labeled images, and QuPath objects. So if you're a Python programmer who wants an intuitive and efficient way to work with QuPath data, you should definitely still be using Paco, although maybe for some things QuBaLab would be useful for you as well. But if you're a QuPath user who wants to dip in and out of Python for some tasks, including a bit of image processing, then you might want to give QuBaLab a try. And I've included a link to the QuBaLab documentation here, which is subject to update as we add some more sort of examples and workflows and maybe update some of the underlying libraries. And we'll be going through some examples that are adapted from the notebooks there and a sort of separate example. And we hope to add some more workflows soon. If you have any ideas for useful workflows, then definitely do let us know. So the first thing that I'm going to do is make sure that I've got some images to work with. So if you download the sample project for the notebook from the repository link that I showed earlier and unzip it in the same directory here as the notebooks, see I've got this i2k QuPath Python project. And then this won't actually do anything, but if you don't have the images, it should download 2 of them for you. So this is going to give us 2 CC0 images. The first one is a CMU1, which probably looks very familiar to any of you who've gone through the QuPath documentation before. And the second one is a CC0 fluorescence image from the EBI, from an EBI dataset. So sort of what I said earlier is that QuBaLab provides a Python API that's quite similar to QuPath's API for sort of for reading images. So we provide a number of provide a number of implementations of an image server, which is really analogous to QuPath image server. QuPath reads images. So here, showing an example of creating an OpenSlide image server, which obviously uses OpenSlide to read RGB images. We also provide a AICS ImageIO wrapper, which will probably be switching soon to BioIO, which is a sort of new version. Oh, good. That's a good start. Not entirely sure what's going on here. I'll visit that later on. I don't think it's important right now. And we also provide ICC profile servers that, if there's ICC profiles available for images, can provide kind of color correction based on the ICC profiles. So in the examples here, we'll be using an RGB image, so we may as well use OpenSlide. So I'll use that server for the next few examples. And so to show some kind of operations you might want to do in an image server, you can query the metadata, which shown here. So things like the number of parameter levels and pixel calibration, also the pixel type and the channel names. Should have fixed this before doing this workshop. I noticed this in i2k. I've got 2 green channels, but that's only because I've renamed the images. As far as I know, it's not bug or anything to be concerned about, but something slightly amusing, I guess. So similar to in QuPath, image server provides a simple API for reading entire images. So here I'm requesting from the metadata the sort of highest downsample or equivalently the lowest resolution image, and I'm asking the server to read a region and just specifying the downsample, which gives me the whole image. And then I can sort of print the image shape and also use pyplot to and a sort of a plot image function from QuBaLab to plot the image. Which, as I said before, if you've gone through the QuPath docs, this will be a very familiar image. And this is a sort of NumPy array. So I guess convenient for lots of downstream workflows. We could also specify XY width and height, as well as downsample to read just a tile of the region, and again print the sort of the shape and plot it. So yeah, here we're just getting our RGB image in a 1000 by 2000 field of view, sort of fairly similar to how you'd request regions in QuPath. But as well as requesting NumPy arrays, we can also request Dask arrays. So this is similar to the example I showed earlier, where I'm reading the lowest resolution image, but here I'm sort of getting a handle on a Dask array, which when it's initialized doesn't have that doesn't actually go ahead and read the pixels until you call compute. So this can be a sort of convenient way to read really large images without necessarily reading all the pixels at once. You can sort of just grab a subset and read those as you need. And yeah, similarly to before, we could request just a tile from the image as a Dask array, and yeah, so here I'd be grabbing the highest resolution version of the image as a Dask array, and then subsetting it in Python before I call compute. So again, this would be a really convenient way of sort of having the whole image as an array, but only reading the parts that you need. If I print and show this, then you can see we've got the same image as before, but this time we went via Dask array rather than by reading the tile from the image server with read region. So far I've been working just in the Python world, but as I talked about earlier, there's a lot of functions in QuPath, or QuBaLab rather, to directly interact with QuPath in a sort of easy to use way. So I'm going to Alt Tab here, hopefully not be too confusing, and I'm going to launch QuPath. So again, here I'm using the version 0.6 release candidate 3, and I launch this. It'll take a short moment. I should have a QuPath window. Good. So I'm gonna first open up the project, the example project that I talked about earlier, which is in this the same directory as the notebooks for me. And then I'm going to go to either this Python logo on the sort of toolbar, or go to extensions, Py4j, and then I'm going to ask it to start a Py4j gateway. And this lets you specify a port, again, because this is communicating over WebSockets, and optionally an authentication token they could use to make sure the connection is secure. So I'm going to press OK, and then I'm going to open this. Oh, sorry, I'm actually not going to open the CMU1 image here. I'm going to open the this HNE TMA image, which is another CC0 image that we've included in the example project. And you can see that I've got a TMA grid specified here, and if I zoom in, there's also a bunch of detections that we'll be working with later, and an annotation. So now if I go back, sorry, I'm going to hit the Alt Tab again. And apologies if my mouse movement is a bit slow. I think somehow Zoom makes my computer a bit laggy. which may or may not be the glorious experience of NVIDIA on the Linux desktop for you. So yeah, now that we're back in Python world, we can use this QuPath gateway method that we've got in QuBaLab .qpath subpackage, and here I can specify the token and the port, which should match what I set up earlier. So this is the default port of 25333, and then I can request QuBaLab to create a gateway using those parameters, and that will mean that we are sort of connected the two processes in, I guess, Jupyter or Python here, and QuPath can now sort of talk back and forth to each other. So not sure why that happened, jumped up a bit. The first thing that we can do is just request a snapshot of the QuPath GUI using this create snapshot, and then I can just plot that using pyplot. And you can see that's captured exactly what's on the QuPath window, and if I move this around, then the snapshot would update. So it's just capturing the current state of the GUI. Yeah. So some other things we can do with this gateway, it's actually an instance of the QPEX class in Java. So if you're familiar with the QuPath scripting API, it provides all those methods that you would be familiar with. So here we can query the version of the Py4j extension that's allowing us to communicate back and forth between QuPath and Python. And we can also ask the current image name, lots of other things. So this is great. We've now got access to the QuPath Groovy API in Python, which means we could, in principle, do lots of cool things. Like here, I'm requesting all the TMA cores from the image, and then doing a sort of list comprehension on this, sort of filtering cores based on whether their classification is positive or not. And then sort of printing the name of the core alongside the number of child objects that has. So if I've got detections within a TMA core, it'll sort of count these and list them out alongside the name. So it's quite nice. Although, as I've noted here, probably for short snippets like this, it's probably quite convenient. But if you're getting to the point where you're writing a long sort of script that's really interacting with Java objects a lot, that's not necessarily something we'd encourage. I mean, for one thing, we don't provide wrappers for Java objects, so finding the right methods can be tricky. You might need to write quite convoluted code, sort of creating a lot of Java objects just to be able to call methods. And debugging will also be quite tricky, as you can imagine, because you're now debugging in two languages at once. And it'll also be quite difficult to sort of maintain complex scripts, especially if things move around within QuPath, the QuPath code base, everything will break. So yeah, if you just want to script QuPath, then you probably still want to stick with, well, you definitely still want to stick with Groovy, and you probably don't want to write sort of overly complex things in Python. Yeah, having said that, we can, as I've kind of shown, and using some other methods, we can request objects from QuPath and get references to Java objects. So here, sort of getting a handle on a sort of equivalent to a Java class here, which is sort of specifying the type of object that I want to request. Here it's an annotation, but we could also request detections or tiles or cells or TMA cores. And then I can request from the gateway all of the objects of that type from the currently open image. So here I've only got one type of, or sorry, one annotation object. So I'll just get a single item, but you could have more. And then I can call this set name method, which is a Java object method on these QuPath objects. And I can set the name to hello from Python, just nice hello world example. And now if I request the QuPath GUI to refresh and take another snapshot, we should be able to see, yeah, there's a name there, which is probably a bit hard to read, but if I Alt Tab, you can see that I've now renamed the annotation object. So brilliant. But as I kind of alluded to earlier, that will be a handle on a Java object. So if you want to interact with it, you're going to have to be calling either Java static methods or object methods, and this can get really kind of convoluted and confusing, and it's not very Pythonic. So we've also provided a converter option for requesting objects. And so what this is doing is basically taking the QuPath object and running it through a converter into GeoJSON, and then giving us a handle on a sort of a nicely wrapped GeoJSON object or GeoJSON compatible object. So if I do something similar before, request all the annotation objects, but convert them using GeoJSON, then I get what a sort of class we've defined in QuBaLab called image feature, which sort of nicely wraps these. And then I could use Shapely or any sort of GIS type Python package to interact with this. Here I'm using Shapely to just show the geometry of the object, but you could, you know, in principle, do all sorts of interesting things in Python. And the sort of nice thing is that because we've got this gateway open, we can convert this object into a pure Python object and then modify it in different ways and then add it back into QuPath. So here, what I'm doing is just taking this annotation object and setting random colors in RGB and then adding it back into QuPath using this add objects method on the gateway object. And if I do this and go back to QuPath and go to my annotations, I've now got a sort of duplicate annotation. One of them is red and one of them is this, I guess, bluish color. So yeah, there's nice ways of requesting objects, and you could transform them however you wanted in Python and then send them back to QuPath in a nice way. And so we've now kind of covered some basics of QuBaLab and interacting with QuPath. So let's show a little workflow that kind of pieces these together. So one thing I didn't show is that as well as these image servers that request pixels from disk, we've also got a QuPath server. Oops, forgot that. That's how Jupyter behaves. So this QuPath server sort of requests pixels from QuPath and passes them through this WebSocket. And so this is really convenient for reading the currently open image in QuPath if you sort of want to do something quickly. But it is worth noting that because we're passing through this WebSocket connection, it's going to be slower than reading from disk. So yeah, here I'm importing this QuPath server function or class rather, and then creating an instance of it, pointing it at the currently open gateway that we have. And this gateway, again, is the communication link between QuPath and Python. And then specifying a large downsample just to make it reasonably quick. And then asking the QuPath server to request the pixels at this downsample level. And so now we've got a nice NumPy array, and we could do sort of whatever we want then using scikit-image and the likes. So here I am sort of converting the image to grayscale and then applying a simple Gaussian filter to remove high frequency content. And then we could do something like importing this threshold otsu method from, against our scikit-image and using this to identify a threshold on our image. and then applying this threshold to the image to create a mask. And then using using another QuBaLab method, we can convert from this labeled image into a Python image feature, which is the type of object that I mentioned up here that we converted to earlier. We can also set the sort of name of the image feature that we're getting out, and then add this back into QuPath. And so I think this will take a little while as it sort of converts between the two types and adds it back in. So see if I can get chat open without crashing everything. So can this be used to train a unit in QuPath? I think in principle, yes. I won't be going through anything that complex just yet, but you know, you could imagine what's the word I'm looking for? Setting up your annotations in QuPath and then sort of reading those regions and supplying them to PyTorch for training. So yeah, it would also be definitely possible to use some of the methods in QuBaLab to read apologies to sort of read annotation objects in Python and then use the image server implementations that we have to read pixels from disk in a sort of nice and convenient way as well. For Mike's question, is it possible to look at the existence of other images in the project, switch the active image, image entry type objects, or anything like that? I actually do not know that. That's a hole in my awareness of the scripting API, I'm afraid. So I believe so, but I couldn't tell you the name of the method, certainly. Great. So we've got our mask annotation created and added it back into QuPath. So before showing what it looks like in QuPath, we can also plot it in Python. So we've taken our image, applied a blur, or sorry, converted to the grayscale, applied a blur, and identified a threshold, and then created a mask from the image and the threshold. This has gotten us a nice thresholded image, which looks reasonable. And if I go back into my QuPath window, you can see that we've got this annotation Otsu object, which again is sort of the equivalent of this thresholded image that we've got here. So I'll just close the chat for a minute so I don't get distracted. Yeah, do feel free to keep asking questions. And so, as I said earlier, we're kind of definitely keen on sort of adding more workflows to these sort of short examples that we've got. Some other ideas of things that might be quite useful here would be maybe creating object classifiers using scikit-learn, or doing object clustering with graph clustering, which I'll show in a minute. You'd also use Python to sort of assess or visualize the feature importance in QuPath classifiers. And as I said before, any sort of ideas or requests that you have, but definitely consider looking into them. So the second piece that I'm gonna go through here, if I go back to my project tab, I'm gonna open this fluorescence image that I talked about, just to start the changes. And so this is I believe a 5-channel fluorescence image. Oh yes, it's really lagging now, which is fantastic. Which just has a single annotation on it. And so I am going to select this annotation and just zoom in a bit, and then open the other Jupyter notebook that I've got in this repository. Start going through this. So the sort of first thing we're going to do here is, as before, we're going to create a gateway, and we're also going to create a QuPath server. So we're going to want to read pixels through QuPath. I've put this in just to sort of reset the state of QuPath, but we won't do anything at the moment because we don't have any detections. So yeah, we've got a nice fluorescence image. We, you know, what you usually want to do in this with an image like this, or often want to do, I guess, is detect some cells. And anybody who's played around with the default QuPath cell detection might know that it can be quite good at detecting nuclei, but especially in images like this, it's not necessarily too great at detecting the cell boundaries. And so just to show again, we can create a snapshot, and this is the current state of the viewer. You've got one annotation selected, and it shows a small region in the middle of the image. And what we're going to do here is we're going to use the instaSeg method that was created by Thibaut in the group for simultaneous segmentation of nuclei and cells using a sort of arbitrary number of channels. So perfect, we've got a 5-channel image here, and we don't have to worry about which channels we're passing into it. So the first thing we're going to do is just import instaSeg and then request this fluorescence nuclei and cells model. You can see that we're able to download it, and also sort of should be able to run it using CUDA. And the next step would be to actually get some pixels so that we can feed it into instaSeg. And so here I'm querying the currently selected object and using the bounding box of that object, sort of the X, Y, width, and height, to read pixels from that image, again using this read region function, and then plotting that. So that should, yeah, give us a nice visualization of what we'll be feeding into instaSeg. And so this is maybe a nice example of although QuPath version 0.6 release candidate 3 currently includes the instaSeg an instaSeg extension by default, which is really convenient for running instaSeg in QuPath. Obviously, a lot of other deep learning models don't have a existing extension, so this is just kind of demonstrating that can be a kind of convenient way to run whatever types of PyTorch or I guess TensorFlow models you might want to use. And so here we're feeding in the image into instaSeg, and instaSeg is going to, oh, sorry, here we're just showing the, oh yeah, so we're feeding the model into instaSeg into this eval medium image, which is going to handle sort of tiling and merging for us. And then I'm going to use a utility method from instaSeg to just show the output. There's a little warning here about pixel sizes, because you should be telling instaSeg what pixel size the image is at. So it's expecting a pixel size of about 0.5 microns per pixel. So yeah, we'll run through on the GPU, and then we'll get sort of two channel output. The first channel here, channel 0, is nuclei, and channel 1 is cells. So we're just going to use the cell channel for simplicity, rather than dealing with both at the same time. And we need to convert these objects. We want to convert this labeled image into features. So instance egg provides us a method to do that, labels to features, which are GeoJSON objects. We're going to select just the channel 1, which again is the cells. And then I've got a little code snippet here, which all it's doing is sort of adding the offset from the original image, because we read a tile in the middle of the image. But instance egg doesn't know that it's a tile in the middle of the image. It thinks it's the whole image. So we're just sort of adding on the offset from the origin to make sure that our detections end up in the same place. So yeah, just again looping through and converting the coordinates. We're also setting the object type to be detection rather than, I think the default is annotation. And then we can add these objects back into QuPath. Oh, interesting. Not entirely sure if that worked. Yeah, it did. Okay, magic. So we've got created a snapshot after adding our detections back in. And you can see, because we added the offset of the annotation, they're all in the sort of correct space. I can press F to remove the fill of the detections, and maybe reduce the opacity a little bit. So brilliant. It's a quick example of running a deep learning model, sort of by reading images through the QuPath GUI and passing them into Python, creating our labeled images and our objects, and then passing it back to QuPath. So that's really good. I want to dive a little bit deeper into these objects. So I'm going to use some methods that are defined in the instance egg extension to sort of add intensity measurements on them. So this will be telling me the average and the variance of the pixel intensities in the different channels. And so I'm using field on the gateway object to access the Java virtual machine, and then sort of diving from there down into sort of Java packages. So here I'm going into the instance egg extension, and then grabbing this detection measure class and making an instance of it, and using that to make measurements on using the current image data on all of the detection objects that we've got currently open. And this is kind of what I was saying earlier, in that you can write fairly complex code mixing Python and Java, but it's probably not a good idea most of the time, because it's going to end up very convoluted like this. Lots of that's a very fragile code, basically, and quite convoluted in that you're always going to have you're usually going to have to create or get handles on Java objects rather than sort of working in the Python world. But for this workflow, it works quite well. So I'll just go ahead, and we can get a list of the measurements that we've got. I'll just print this at the end. So yeah, we've got a whole list of measurements. I can then filter this just to include the mean. So here I'm just looking at the mean DAPI and these opal channel intensities within the cells that I've detected. And then I can sort of collect these into a a pandas data frame, and then I'm going to just standardize them just to avoid effects from sort of overall scale and shifts, which is probably not going to be terribly interesting. And then because we've got a pandas data frame of all our objects and all the measurements that we made, these intensity measurements, we could now run a dimensionality reduction algorithm. So here I'm using UMAP, but you could use any sort of fancy autoencoder type method. And then I'm looping through all of these measurements and then adding them, or looping through the embedding that we get from UMAP and then adding them back into the QuPath object measurement lists. And so that might be useful if you, again, say use something like an autoencoder to sort of compress your data in an interesting way, that you might be able to use them in an object classifier after the fact, or visualize them using interactive plots in QuPath. But we're going to stay in Python world for the time being, and again work on this normalized data frame of measurements and just compute the k&n graph and then convert that into a sort of an adjacency matrix and adjacency graph. And then we could do something like cluster the k&n graph. So here I'm using Leiden to find a partition of the graph based on the graph modularity. And then again, I'm looping through all these objects and setting the classification. So here I'm just saying, cluster 0, cluster 1, cluster 2, etc. So hopefully this should run pretty quickly. I'm told there is a substantially faster version of a lot of these algorithms implemented using GPU acceleration, but I'm not showing that at the moment. But yeah, if we again refresh the QuPath GUI and take a snapshot, we can see, or at least very faintly see, because I lowered the opacity and removed the fill on detections. But yeah, if I add those back in, you can see that we've clustered the cells. And if I press D to show and hide these, you can see that it's done something at least vaguely sensible. And that, like, the obvious, there's some obvious categories of cells that seem to correspond to the clusters we've made. Although obviously I've not done this in a terribly rigorous way. You'd want to do all sorts of things like and so I alluded earlier to that you could use this stuff in QuPath. One example that you might want to do is this quick plot script that I've included in the repository as well. So what this is doing is creating a scatter chart of the measurements of the detections. It's going to be linked to the viewer so we can interact with it, just setting a little title to describe what it is. And then I'm setting the X and Y as these UMAP1 and UMAP2 measurements that I added back in using the QuPath gateway, and then just showing it. So this should pop up a little window. Good. I was a bit nervous when it stalls like that. And this is showing us the all the detection objects colored by whatever cluster we've made. As I kind of said, you probably would want to spend a lot more time on making sure this is real if you were doing it in an actual analysis, because this partition of the cells is probably a bit arbitrary. But one nice thing about this plot being interactive is you can click on individual points and it'll show you them. So would be one nice little workflow. Cool. So I'm gonna open the chat now. I've gone through all of the material I was hoping to go through, and hopefully there's good. So Pete, as usual, is able to answer what I wasn't regarding the image entries and projects. So Mike's question about creating multiple socket connections and connecting different Python instances that do different things to QuPath. Interesting. So at the in principle, I think you could. Whether or not we currently allow it is a different question. Certainly, you could. I think you could communicate from two different Python processes to the same gateway without any issue in terms of creating multiple gateways on the QuPath side and communicating to them individually. I don't know if we allow that, but I think in principle you could do. Um, I yeah, I don't know how fun it would be to have multiple things communicating on the same gateway and doing things. Certainly, if you try to send two things at once, bad things might happen. Um, and I think there's probably going to be a trade-off, as you say, between reducing dependency hell in Conda environments and not making a different type of hell for yourself by trying to figure out which Python session the thing that you want to send is in, and yeah, all sorts of messiness like that. Good. Yep. Definitely recommend checking out Thibaut's workshop. It's a really nice and easy to use Python package. That the method is really, really exciting as well. I found it really, really useful. Can you do some segmentation and store a GeoJSON file using QuBaLab? Yeah, so the GeoJSON file you could use you could write that to disk in JSON format and definitely read it back in either in QuPath or read it back into Python or sort of any language really that supports GeoJSON or can parse JSON objects. Yep. Pete's definitely on the ball with the sort of the aims of QuBaLab and what we've implemented so far. How many cells can the plot in QuPath reasonably handle? Not that many at the moment. If you got into millions, you're probably gonna want to plot a subset. We're sort of considering options for how we might be able to support more objects in future, whether that's using a different type of plot or sort of implementing some smarter subsampling strategies where maybe you're not plotting all of the cells in very dense regions, but more plotting all the outliers and then subsampling points in the middle, or using a more efficient plotting technique or library that should be able to handle more objects. I unfortunately made some promises that I don't know if my uh, don't know if my coding can support that I would get this implemented in 0.6, but we will see about that. Yes, being able to UMap across images in a project and jump to the image and the cell, that would be indeed very useful. Currently, it's the sort of viewer interaction is only jumping within an image. So that's a sort of second level request that or a second level feature that might be more difficult in terms of jumping images. It would also probably test your patience as you have to wait for an image to load and the viewer to jump. But So the current notebook is clustering objects. So I don't think I've missed anything. If I have, then do feel free to um, shout. And yeah, if there's any other questions, then please do keep them coming. Okay. Um, Examples of sending generic information back to QuPath, text like listing positions. I'm not totally sure what you mean. Um, yeah, that's kind of the question I was asking, like, well, you know. Ah, um, not totally sure. I'd probably have to have a concrete example of like what type of information you want to send and where it might be the best to put it. But yeah, you can definitely. Ah, okay, so things like metadata. I believe that you can update the uh, image metadata, um, sort of adding arbitrary key values stuff there. That would be one place to put it, I think. Uh, Position of tiles maybe sounds like a object thing. Object thing is a bit vague. Might be best using QuPath objects like annotations or tiles or something similar. But yeah, definitely sounds like it would be easier than um, writing to a text file and reading back. Yep, definitely feel free to um, ask questions on the forum or uh, yeah, reach out directly if you have any specific um, sort of questions or feedback about this. And yeah, hopefully we'll have QuBaLab on PyPy relatively shortly and um, QuPath 0.6 out soon TM once we've got everything sorted out. So okay, I think I'll call it there. Thanks so much for coming and hope you found it useful. And yeah, cheers, bye.